cell division, mitosis, and cell transfer mechanism. In this video, we will be demonstrating to you the procedure steps found in body, demonstrating to you the procedure steps found in body, using digital art, illustration, and animation. We hope you enjoy this video, and we hope you enjoy this video.
Before we begin, let's define some important terms starting with mitosis.
So I was in biology class, and mitosis. Oh my god.
Mitosis is a type of eukaryotic cell division that produces two identical daughter cells. It includes four major stages: prophase, metaphase, anaphase, and telophase. Now let's move on to the procedure for the experiment. To start, we examine a digital representation of an onion root tip. Under the microscope, we can observe the different phases of mitosis. In prophase, the chromosome starts to appear clearly. In metaphase, they line up perfectly in the middle of the cell. In anaphase, the chromosomes get pulled apart towards the opposite sides. And in telophase, two new nuclei begin to form. Wow! It's amazing that we can actually observe these different stages. This helps us clearly distinguish each stage of mitosis.
Now let's move on to the next part of our experiment, which we call cell transport and mechanisms. Let us first define cell transport and its mechanism.
Here is cell reproduction explained by tiny cats. Time for a meowtosis miracle. One tiny cat magically makes two. First, our tiny cat needs to copy everything inside it. It's like making a perfect recipe twice. This copy is called the DNA, the blueprint for being a cat. Then the DNA doubles and gets ready to separate, like two balls of yarn that need to be untangled. The cell grows bigger so it can make enough space for the division. Now, the DNA copies move to opposite sides of the cell. The cell starts to pinch in the middle, like a balloon animal being twisted. Finally, the cell splits into two new identical tiny cats. Meowgical! Meiosis in a nutshell. Copy, grow, divide, repeat. Two tiny cats for the price of one.
First, cell transport is the movement of substances across the cell membrane. This process helps cells maintain balance, or what we call homeostasis. Now, we will demonstrate the four different setups in this experiment. The first setup, the setup A, which we call osmosis in raisins. For setup A, we explore osmosis using raisins. First, we place 5 dried raisins into a beaker that's half filled with water. We let the raisins stand at room temperature, and after a while, something changes. Wow. We observe that the raisins swell as water enters them through osmosis. Pretty cool, right?
Wow! Ang galing! Galing mo naman!
Next, we have setup B, the effect of stirring on dissolving sugar. We prepare 2 beakers of water and add half a teaspoon of sugar to each. In the first beaker, we stir until the sugar fully dissolves, while in the second beaker, we don't stir at all. We then compare how fast the sugar dissolves. We observe that the stirred beaker dissolves sugar much faster because stirring increases molecular motion.
Now we're on to setup C, the effect of temperature on diffusion. We prepared 2 beakers, one with hot water and one with cold. We add 1 teaspoon of coffee each granules to each and let them stand. We observe that coffee granules spread much faster in hot water, showing that higher temperature makes particles move more quickly.
Last but not the least, setup D, filtration. We mix charcoal, copper sulfate solution, and boiled starch with distilled water. After observing their characteristics, we pour the mixture in a funnel lined with filter paper. We then examine what passes through the filter and what stays behind. Here are the results. Charcoal stays on filter paper because its particles are large. Copper sulfate passes through and appears in the filter. Starch does not pass through when iodine is added. No purple color forms, which means starch is absent in the filter.
At this point, filtration is mostly complete. Looking at the solution, it's clear that now the charcoal has passed through the filter. I will now test the filtrate for each of the three possible solutes, beginning with the chloride ion. Here, I carry out a test for chloride in the silver solution. Next, I test for starch using the iodine solution. And last, I test for glucose using Benedict's solution. I take the test tube to the hot water bath for it to be warmed up for 5 minutes. 5 minutes later, you see that a change has clearly occurred.
And that wraps up our virtual laboratory activity on mitosis and cell transport mechanisms. Through digital illustrations and animations, we explored how cells divide and how substances move across cell membranes through osmosis, diffusion, dissolution, and filtration. Thank you so much for watching and listening. We hope you enjoyed learning with us. Have a great day. Bye!
Yes, finally! I can rest.